Thank you, Josephine, and thank you to the organization for the opportunity to present Capricor Therapeutics. I joined Capricor just over the summer, and I'm going to share with you a little bit of where we are as an organization. I'm going to make some forward-looking statements, so that's our disclaimers. So Capricor is a cell therapy biotech company based out of San Diego. We have manufacturing capabilities for clinical trials in Los Angeles. And we have two main programs or platforms. One is focused on the treatment of Duchenne muscular dystrophy. We announced a partnership in the US with NS Pharma, who is already in the field. This partnership is helping us fund the phase 3 trials. And we also presented phase 2 data earlier this year in the Lancet, and more recently the open label extension of this OP2 phase 2 trial at the parent project muscular dystrophy back in June. The second platform is an exosome-based technology. I'll cover a little bit on that. And all this is anchored in robust science with a lot of patents that we got from Cedars-Sinai or Johns Hopkins, and some patents that we developed on our own, obviously, as well. We currently have cash runway until Q2 2024. This is our pipeline. Again, the most advanced asset is CAP-1002, phase 3 clinical trials that we started enrolling in July, as we announced at our last earnings call. Other programs are in various preclinical stages. I'll touch on a couple of them. So at the small organization with two platforms, as you can imagine, there are a lot of exciting things we could do. Just because we could, though, doesn't mean that we should. And there are, however, a few things that we must execute on in the coming months. First and foremost is our Duchenne muscular program. We are in phase 3. That's, as you all know, very exciting, but also very involving. We're actively engaging with the FDA under our Avat designation. And right now, we're also pursuing OUS or partnerships outside the US to commercialize this asset. We're not planning on going direct, obviously, outside the US, and we've already partnered in the US with Nippon Shinyaku. I'll touch on our StealthX platform with two main applications that we are developing at this point, and then I'll give you some corporate updates and milestones in particular. So most of you are probably familiar with Duchenne muscular dystrophy. It's a genetic defect affecting mostly young boys who cannot produce dystrophin or produce optimal dystrophin or non-functioning dystrophin, which leads usually to the muscle degeneration and inflammation, and eventually to muscle cell death. And this is a highly debilitating disease with a high burden on patients and a life expectancy for these young boys in the 20 to 25 years of age. Our therapy is a cell-based therapy, cardio-derived cell-based therapy that is functioning through the secretion of exosomes. These exosomes go into the cell muscles and release their cargoes, in particular microRNAs. There are a few of these microRNAs that are in particular very instrumental in reducing the inflammation within the cell muscles and also have pro-regenerative functions. So this is the core mechanism of actions of our cell therapy. And we have received several designations from the FDA, in particular the RMAT one, that is allowing us to stay very engaged with the FDA and also potentially could support a rolling filing. This is a population we are targeting, boys aged 10 and or older. At what point most of these boys become non-ambulant. This is about 10 to 15,000 patients in the US, so the prevalence pool is quite large, with an incidence of about 1,000 to 1,500 every year. And so our clinical program is focused on these boys 10 years and older, when most of them lost their ambulatory capabilities. This is a phase 2 data that we presented earlier this year. primary endpoint is the performance of the upper limbs for these patients. Again, most of them are getting to a non-ambulant stage, so the primary endpoint couldn't be a walk test, for example, but we're measuring their ability to use their shoulder, forearm, and wrist. And our phase 2 data showed a significant slowing down of the progression of the disease. It's not a cure, but it's a significant reduction of the progression of the disease. And also as secondary endpoint, very interesting as well, was the preservation of the ejection fraction in these patients, which as they progress, usually, these cardiomyopathies are what leads to the extension of these patients. We more recently presented our open label expansion. So we've given the opportunity to these kids who were on the OP2 trial to get on our open label program. And we presented the 12 months data in June. We're now continuing to collect data. I've collected now 18 months, and we'll announce that probably later this year. But what's interesting and important to see with this open label extension is the potential durability of this therapy, which is, of course, very material for patients and also material as you build a franchise for an organization. And the other important or potentially, significant element here is the potential disease-modifying effect of this therapy, because what we see is that kids who were on therapy, when they got off therapy for this gap period that we have in the middle here, they didn't lose their gain, if you want, or they didn't go back to the placebo arm. So that's real functionalities that they preserve over time. And I like to say that the area between the two curves here are tangible functionalities for these patients. That's very meaningful for them. It can be the difference between being able to drink by themselves or need help to use a glass of water. And so because of this mechanism of action, we believe that CAP-1002 has the opportunity maybe to become a new treatment paradigm for these Duchenne patients and potentially become a backbone therapy the same way that steroids is being used right now. And we see our therapy potentially also being synergistic with a lot of the other therapies that are under development. These kids need all the therapies that they can get their hands on. And so we really also look forward to potentially doing some combination therapy evaluations. So this is our phase 3 trial that is currently enrolling US-based only, targeting 20 sites. Back in September at our last earnings calls, we were about 6 weeks into the beginning of the phase 3. We had activated 4 sites and enrolled 7 patients. So there is a lot of expectation for this trial. The design of this phase 3 is exactly the same or very, very similar to our phase 2 design, working closely again with the FDA on this. And the primary endpoint is going to be the change in this performance of the upper limbs of patients, with a secondary endpoint on the cardiac and QOL. So this is our core program, and where the company is mostly focused, as you can imagine, 65, 70 people in the organization, a phase 3 trial, that's very demanding. But over the years, and also because our cell therapy works through the expression of exosomes, we've become more and more knowledgeable and have invested also in an exosome platform. And what I'm going to describe today is the progress we've done in 2 applications for this exosome platform. So maybe just as a way of background, exosomes are widely characterized and known. They are the cell signaling systems, and so produced by pretty much every cell. And each time we receive a transfusion, we probably get billions of exosomes. So their safety profile is also pretty robust. And so what we've done over the years is focus on 3 main areas for this exosome platform. We've heard a lot through this conference about the challenges or potential challenges about manufacturing and producing cell therapies in a reproducible, scalable, predictable way. We invested a lot of time on that. I'll touch on this in a minute. And then our platform is leveraging two two aspects of exosome technologies, the loading of these exosomes and the targeting of these exosomes. And we're doing that through what we call StealthX, which is our proprietary fusion protein and linker approach, and that has allowed us right now to develop this platform for two main applications in the vaccine space and for therapeutics, targeted delivery of therapeutics. So I mentioned manufacturing, and here, while none of these steps in themselves are maybe necessarily, very specific to Capricor, the combination of these different steps is where there is a lot of know-how. And also probably as important, and we just heard that from the FDA in the other room, the characterization and the quality control of these exosomes is also very important. And so we've developed, largely in collaboration or at the request of the FDA, a lot of assays on our exosomes, in particular potency assays. So when it comes to loading, there are two main ways of loading exosomes. You can use physical approaches, which have been around for many years, and there is maybe next generation's physical loading. But where we are more focused at Capricor is our endogenous loading technology with StealthX, as I mentioned earlier, where we basically engineer the producer cell lines to have them produce the cargo of interest, or to have them attract the cargo of interest within the cells, within the lumen of the exosomes, or to express these cargoes, these loads, on the exterior of the exosome. And so, as you can imagine, when you start expressing, let's say, a protein on the surface of exosomes, these proteins could be used as vaccines, for example. And this is our vaccine platform where, and it's a very, here it's simplified, obviously, but in a nutshell, you select the antigen of interest, you engineer the producer cell line, and then you produce exosomes that are going to become your vaccines for the given antigens. And so this is a very versatile platform. We work with very rapid cycles, similar to mRNAs. We don't have any adjuvants in this technology. And yet we've demonstrated that we're eliciting very, very strong immunity. We presented our first data on this platform last week, in fact, at a Boston conference. The other aspect is targeting, very exciting as well, where you, again, use the same technology to express a protein or a vector, targeting moiety on the surface of exosomes. And these targeting moieties are selected based on their affinity to biomarkers on tissues of interest or cells of interest. And then you can load the exosome with a cargo of interest. So now you have targeted vehicles for precision therapeutics. And this is the second leg, I would say, to our exosome platforms, where preferably we're starting, in fact, with validated cargo so that we don't introduce the viability of a novel cargo or novel molecule. And we engineer these targeted exosomes to go to select tissues. So, we've heard a lot of this targeting works done with LNPs, and exosomes are just another way, we believe a better way, to build targeted vehicles. They have not seen, of course, as much clinical development as LNPs, but they don't also bring some of the safety challenges that LNPs are known for. So the last part of our prioritization and strategic focus is more at the corporate level. So I mentioned manufacturing. This is, in our space, something that is very time-consuming and a strong focus of us because we know that cell therapies obviously are not antibodies or small molecules. We've invested and are continuing to invest a lot, in particular in process development, process improvements, so that we can, when the time comes, easily or more easily do tech transfers to CMOs, for example, if needed, or scale up in our own manufacturing site if this is the path that we were to choose. And we're also looking at partnerships. So I mentioned The Duchenne program, where we currently have a partner in the US that is fully equipped to launch this product in the most important market, obviously, but we are now actively looking at partners for Europe in particular and Japan. On our targeted delivery platform, we already have conversations with companies, but we're going to decline, if you want, or build this technology toward multiple type of tissues. So we're starting with just one tissue. We've not announced that yet. We're probably going to do that at our next earnings call. And then from there, we're going to take a very similar standardized approach to go after other tissues of interest. And then last but not least, our vaccine platform. You've probably heard this morning in the space of Exosome, news from some of our colleagues on the East Coast. And this is an area that is very active, where we believe we have a template approach, a rapid development approach, protein-based, that could potentially compete with mRNA vaccines. So stay tuned. Lots of upcoming milestones for us. In particular, next year, middle of next year, we have our interim review of our OPS3 data. We think we will have fully enrolled by Q3 of next year. And these are some of the other milestones. We believe we also have the team to execute, and executing in a small organization like us is very important. And with that, I want to thank you for your time.